For this little short lecture, I want to talk a little bit more about the genetics of human traits. I'm going to start out with another non-disjunction example, which should be a little bit easier now that you've all thought about non-disjunction and Down syndrome already pretty carefully. Then I'm going to talk about a few traits that are difficult to study and some of the difficulties in studying them. And then in a separate lecture, I'm going to talk about just a few more human traits that are interesting. So let's start out with some more non-disjunction. Each human somatic cell normally has 46 chromosomes. These are the non-germline cells. In meiosis, they'll form the 23 pairs. Now, 22 of the 23 pairs are perfectly matched in both males and females. Oh, sorry, I have a fan going on. It's blowing my papers everywhere. So these are 22 out of the 23 pairs. These are perfectly matched in males and females. So those are all of these ones here. And we call these the autosomes. Auto, like an automobile, somes. And one pair are the sex chromosomes. And human females and mammals in general are XX and males are XY. So here is a human female who is XX. The genes on the Y chromosome are the initial trigger for maleness, so for the phenotypes that we associate with being male. But sometimes errors occur during meiosis, and non-disjunction is the failure of chromosomes to separate correctly during either meiosis 1 or meiosis 2. So here we have non-disjunction in anaphase 1, and you can see that the products of meiosis 1 are going to be different from each other, right? So these two cells have this is after meiosis 2. These two cells have one too many chromosomes, and these two cells have too few chromosomes. And we call this aneuploidy. So aneuploidy is when a cell has an abnormal number of chromosomes. And most of these kinds of non-disjunction abnormalities just lead to a failure to develop or to early death before adulthood. But individuals with an extra copy of chromosome 21 or chromosome 22, which we've talked about in terms of Down syndrome, can survive to adulthood. But these individuals will typically have some delayed developmental and mental impairment. Down syndrome, that's the extra copy of 21. So here's just a karyotype of someone with Down syndrome, and we've already kind of gone over this. This small chromosome here, this is 21, it's really, really tiny compared to other chromosomes, and compared to other genes, it doesn't have a ton of genes on it. So an extra copy of it doesn't always lead to really huge phenotypes. And so you can see that in this guy here who looks like pretty indistinguishable from someone who doesn't have this extra chromosome copy. And we also know from Down syndrome that the incidence of Down syndrome increases with higher maternal age. So after the age of 35, the incidence increases from about 0.2% to over 1%. So why is this? Well, we know from meiosis that a cell can get an extra chromosome when non-disjunction occurs. So the meiotic spindle kind of pulls an extra chromosomal copy into a cell, and the other cell doesn't get that, right? So it gets lost from it. So in the case of chromosome 21, the cell missing a copy will just die. Like there's no way for it to go forward. And the cell with the extra copy will produce an embryo with Down syndrome. In humans, egg cells remain arrested in the prophase of meiosis 2 until they're fertilized. And so that means that their chromosomes are in synapsis for years. So human females are born with just about all the egg cells that they'll ever make. So after about 35 years of sitting in synapsis, the synaptic proteins start to actually form covalent bonds with each other. And we call this cross-linking. So in a way, you can think of this as just these chromosomes become more stuck to each other. They become stickier. And so these extra covalent bonds mean that the chromosomes end up sticking together as the spindle is trying to pull them apart. So it's more likely for two of them to get into one cell. Most of these non-disjunction embryos die just soon after fertilization. And this actually partially accounts for the very high early miscarriage rate we see in women over 35. This makes up a large portion of the lowered fertility in older women. Also, older women tend to have fewer eggs, and so their ovulation times, the distance between their ovulation times can start to increase as they get older. Most of the non-disjunction embryos die soon after fertilization, like I mentioned, but if you only have an extra copy of chromosome 21, these embryos will often survive. And this explains why the rate of Down syndrome is so high in older mothers. Oh my gosh, excuse me. The smoke from the fires is really getting at my system. I've got this like crazy sore throat and my eyes are all red. Okay, I hope you all are doing a lot better through all these awful fires. All right, so that's the non-disjunction case that we've already thought about, but I want to think about another set of non-disjunction cases, which is when non-disjunction affects the sex chromosomes. And so a really important thing to know is that for mammals, only one X chromosome is pretty active. So if you're an XX female, you have two X chromosomes, only one of these is going to actually make very much product. The other one's a little bit active, but not super active. And we call the extra X chromosome a bar body, and it gets deactivated really early on in development. And what this means is that if you have extra X chromosomes in your body already has a mechanism for dealing with those extra X chromosomes. And if you only have one X chromosome, your body's perfectly happy, right? So males typically only have one X chromosome and they're like perfectly fine and alive. So a female with only one X chromosome still has all of the instructions that she needs to build a body. So I want to think about non-disjunction of this X chromosome, this sort of weird chromosome that most females already have sort of this sort of extra copy of compared to males. So this is, we get non-disjunction of the X chromosome gives three possible conditions. So one is a triple X female, and don't Google image search for that one. I made like a terrible mistake. So these triple X females, it's also called Trisomy X. They're usually taller than average, but other symptoms vary. Like there's no like one set of things that happens if you have an extra X and you're a female. Usually you're like pretty normal. You could also be an XXY male, and that's what we see here. This is a male with what we call Klinefelter syndrome. So he has his regular X, his regular Y, and then he also has an additional X. Now, these males look like really normal. They do tend to be sterile. They're almost always sterile, and that's just because their cells can't really undergo meiosis because they have this extra chromosome hanging around that mucks up the works. They also do have they're not really good at growing beards typically. They tend to have a little bit of breast development, but it's pretty minor. They will often have slightly smaller testicles, and then what we call the female pubic hair pattern. I don't know if you've had a chance to check out typical male versus female pubic hair patterns, but males tend to have more dispersed, sparser pubic hair, and females tend to have like this like tighter, like tidy triangle of pubic hair. That's like the typical pattern. It's obviously not true of everyone. So again, mostly pretty normal looking dude with a couple of like little minor funky things. And most guys who have this don't ever find out they have it unless they're trying to have kids and they're wondering why they aren't able to get someone pregnant. And that's because their cells are all like confused when they try to undergo meiosis with this extra chromosome hanging around. All right, so here is our last non-disjunction of the X chromosome, and that is with a Turner syndrome female. So she's an XO. When we say O, we don't mean she has some special round chromosome. We just mean nothing's there. So she has one X chromosome and nothing that can pair up with it. So again, same idea with her undergoing meiosis. She has an odd number of chromosomes, so they can't match up. And so she ends up being sterile because her cells don't know kind of how to figure out meiosis for her. She's also super, super normal looking. She tends to be a little bit short. She tends to have some webbing on her neck, which is something that if someone has Turner syndrome, this usually is surgically removed when they're really young and like nobody ever sees it. And then also she tends to have a slightly broad chest. So again, like not something that you would really notice as being out of the ordinary. Her ovaries do not grow well, and this might have to do with her estrogen levels being somewhat low. And then she also tends to have like lots of little freckles. So again, almost completely normal with just these extremely, extremely minor phenotypic differences from a standard XX female. So here is just another picture of a male with Klinefelter syndrome. And you can see this guy looks like, you know, kind of any dude you'd see with his top off at the beach. I don't know why he's in his bedroom. I love stock photos. They're like pretty amazing. or stock pictures. So he just has this extra X chromosome. So typical males are XY, and he has this one extra from non-disjunction. So here's a Trisomy X female. Again, so she has an extra X chromosome. This is before replication. In this picture, we see replicated chromosomes. So these are non-replicated chromosomes. And she just tends to be tall. Like, that's really kind of it. There's like not a huge phenotype associated with having an extra X chromosome. So let's just see really quickly the non-disjunctions that create too many or too few copies of the X chromosome. So here we have our Punnett square. And we've got these this non-disjunction in this one female. And so she either produces eggs that have one too many X chromosomes or no X chromosome at all. So one of her eggs got 2 X chromosomes, one of her eggs didn't get it during non-disjunction. So she is going to mate with like a standard male. If he undergoes non-disjunction, it's a slightly different story. And we can kind of think about that in a minute. So if she mates with a normal male, then we can get fertilized eggs, so embryos that either have 3 X's. They got the 2 X from the mom and 1 X from the dad. If you get the 2 X from the mom and the Y from the dad, you're a male with Klinefelter syndrome. If you get this O egg with an X from the dad, you have Turner syndrome. And our only non-viable option is if you have no X at all. So that's if you get a Y from your dad and no sex chromosome whatsoever from your mom. This is non-viable. The Y chromosome is actually really small and it doesn't have the genes you need to make up for a missing X chromosome. So again, we're going to get a son with 2 X chromosomes, that's Klinefelter son, a daughter with Turner syndrome, so that's just 1 X chromosome, or a daughter with 3 X chromosomes, and the OYs are inviable. So like I mentioned before, you can also get non-disjunction of the Y chromosome. And here is an example of this. So in these cases, you get YY gametes in a male. So the sperm will actually have 2 Ys in it. And so if that fertilizes an egg, that's going to give you an XYY male. These males are fertile. They're 100% normal. We have no idea how prevalent this is in human populations because there's like no phenotype associated with it. So we just have like no clue how prevalent it is. Like half the people on the planet, well, they don't. Half the people definitely don't have it, but we don't know if it's like 0.0001% or 0.01% or what, because they're not sterile. They're fertile. They have totally normal appearances. And unless someone gets genetic testing, they probably will never know that they actually have it. All right, so I want to move into some other genetic traits. So these are ones that are not caused by non-disjunction. These are ones that we typically think about as a little bit more Mendelian. Right, so why are human traits so hard to study? Well, they can be really complex, meaning that they have a lot of factors factoring into them. Lots of genes plus environmental factors, and that can make them really hard to study, especially because humans themselves are complicated, right? So if I want to know where my big nose came from, I can't just say, like, what's the big nose gene, right? Because there isn't going to be one, right? What's the actual shape of my nose? How big is it? Is it super big or just kind of big? It's super big. And is it big in kind of what ways? So that's a complex trait. It's also hard to study humans because we can't crossbreed humans. We have to rely on like family pedigrees to think about like who mated with who. And also a lot of interesting traits just aren't medically relevant. And that means it's really hard to get funding to study them. So here's one example of this. This is a widow's peak, which someone in the discussion board brought up. So here we have a widow's peak. Here's someone without a widow's peak. And the question here is widow's peak a dominant or recessive trait? So let's look at this family pedigree to try to figure this out. Here's our first generation, the grandparents. And so we have someone with a widow's peak mating with someone without a widow's peak in 2 different generations. So here are their kids. In this family, we get about half the kids having a widow's peak. Same thing in this family. So now we have 2 people with a widow's peak having kids to each other, and we see that they make 1 kid with a widow's peak and 1 kid without. Now that tells us that widow's peak has to be a dominant trait, because otherwise you'd never be able to have people who both have a widow's peak producing children who don't have a widow's peak. Okay, so there's 2 issues with this. So one is check out your own hairline. My hairline is like jagged. I've got a messy hairline. So Do I have a widow's peak? Probably not. But you can also tell looking at people's hairlines. Some people have a very pronounced widow's peak like her, and some people have kind of like a more subtle widow's peak. So it's clearly not a simple like yes, no trait. The other problem is when people made this kind of a pedigree, they really only looked at one family. So we don't have a lot of scientific studies on this. What happens if we look at other families? Do we see the same pattern or do we see a different kind of pattern? No one's done this yet. Are you going to be the person who does the first like real scientific widow's peak genetic study? Maybe. So same deal with attached earlobes. So here we have an attached earlobe situation where we see a family where this mating pair produced, which have free earlobes, both of them, produced children with attached earlobes. And so these attached earlobes are just like where you attach it here. And so this suggests that attached earlobes is a recessive trait because otherwise this mating would never produce attached earlobes. Okay, and same thing with this mating too. So we have two people with the dominant trait producing people with the recessive trait. So it must be this must be recessive. But again, this is just one family, and what happens if we look at a lot of other families? Now, someone actually did this time, so you don't have to do it yourself. Researchers did this with a bunch of New Guinea families, and the yellow is what we would expect if attached earlobe is actually recessive. So if you have parents who have free earlobes, we don't know what their genetics are. If they're both dominant homozygotes, all of their offspring would have free earlobes. If they're both heterozygotes, then some of their offspring would have attached earlobes. We don't know. Same thing with this situation, attached mated with a free earlobe. We can't really tell what it would be because we don't know if this person is a heterozygote or not. But on the other hand, we do know that if we have two individuals with an attached earlobe and attached earlobe is recessive, that means both of these individuals would have to be little f, and all of their offspring would have attached earlobes. Okay, so none of their offspring would have free earlobes. So here's what they actually saw. They actually saw that if you have two parents with attached earlobes, you still have a 15% chance of having free earlobes. So that means this is not a simple Mendelian trait. It's pretty clear that there is a genetic component with this, right? Because we see that parents with free earlobes tend to have children with free earlobes. Parents with attached earlobes tend to have children with attached earlobes, but it's not as straightforward as if this were strictly Mendelian inheritance. So we assume that there are actually multiple genes involved in this. And again, a lot of human traits are complex like this. They're determined by multiple genes as well as environmental components. So for example, you might notice that skin color is similar within a family, right? So if we have two families with pale skin, they tend to have pale skin children. If we have two families with darker skin, they tend to have darker skin children, right? So we know there's a genetic component to this just looking around with our eyeballs. But your skin color probably isn't exactly in the middle of your parents' skin color, and it probably isn't exactly like either one of theirs either. And that's because there are a lot of genes that influence this. And your own skin color also depends on your health and how much time you spend in the sun. All right, handedness. This is one of my favorite ones because this is a really complex trait that people are like really obsessed with. I mean, I am because it's one that it feels like so easy to track. And so you kind of get in your head on it a little bit. So this is my family looking at handedness. And so both of my parents are left-handed. And so here's me. And I have one left-handed brother and one right-handed sister. Okay. Now here's my spouse's family. And both of his parents are right-handed. And he's also right-handed. So we have two right-handed people now having kids. And we have two sons. One is right-handed and one is left-handed. Okay, so this is like kind of weird because if this was recessive, we would expect my parents to have all left-handed children. If it was, and we really don't see that. If it was dominant, then both of these people would have to be heterozygotes. They would have to pass down the heterozygosity to him, and I would have to be a heterozygote in order to see these left-handed children. And that's totally possible. But if we kind of zoom out on the whole picture, that's not what we see. What we do see is that there's a genetic component, meaning that left-handed parents tend to have left-handed children, right-handed parents tend to have right-handed children, and left-handed is more rare, but it's most likely determined by multiple genes as well as environment. For example, My grandma swears that she was born left-handed, but her teachers, she went to Catholic school back when you could still like hit children with rulers. She said that the nuns beat it out of her. I don't know how true that is. This is my grandma's story. She's a little like, she likes to tell stories sometimes. But we know that environment plays a factor. You can force yourself to learn to write with your non-dominant hand. We have a similar situation with eye color, though the eye color situation is more well understood. It's really obvious that eye color is genetic, right? If you have blue-eyed parents, you're more likely to have blue eyes than if you have two brown-eyed parents. And also unusual eye colors like ice blue or violet, these tend to run in families. But there's so much variation in eye color. This is just looking at some of the variation in eye color, that it's really obvious immediately that it can't be a simple Mendelian trait. So here's eye color in my family again. So here is my spouse, and his parents both have blue eyes and he has blue eyes. Here is me, and my parents, one has brown eyes and one has green eyes. My mother, who has green eyes, got her green eyes from her parents. She has a blue-eyed mom and a green-eyed dad. My dad got his brown eyes from his parents. Both of his parents have brown eyes. And I'm assuming that everyone behind here also has brown eyes, so I don't have pictures to prove it. All right, so my parents, one with brown eyes and one with green eyes, had two brown-eyed daughters and one hazel-eyed son. I tried to make this hazel. I was like a little bit of a tricky one to do. So his eyes have some green around the middle. Wait, no, some green around the edges and some brown around the middle. Now, unfortunately, he's also our lefty in the family, and I wish that he would just go produce offspring so I can see what some of the underlying genetics are to this, but he refuses. He doesn't really want kids. So we're never going to know what the genetic components of this are in my family, since the one interesting progeny refuses to pass on his genes. Now, my sister and I are uninteresting. We're both right-handed and we both have brown eyes, and all of our children also have brown eyes. So hers is not a surprising case because the dad also has brown eyes. Mine, it's maybe not surprising, maybe a little surprising. The dad also has blue eyes. But again, I'm inheriting some amount of blue-eyed genetic material and green-eyed genetic material from this side of my family. So basically we have this sort of like brown lineage here, kind of winning the day with all brown offspring in this generation here. Now, eyes, we know a lot about how the pigment is regulated in eyes, and there's one kind of simple pathway that accounts for a lot of the eye variation that we see, basically the brown versus blue decision. Now, once you have sort of these kind of like bluer eyes or browner eyes, so one or the other, there's a lot of downstream stuff that modifies that. Are your eyes dark brown? Are they light brown? Are they more yellowy brown, or are they more blacky brown? Your blue eyes, are they green or are they blue, or are they kind of in the middle? Are they hazel? And so this is looking at this OCA2 pathway. So OCA2 is a protein that causes the generation of pigmentation. So you can have brown pigment or like no pigment, and that's blue eyes. They are just missing this particular kind of brown pigment. They have their own pigments in them, but they don't have this one pigment that's made by OCA2, the pigment that gives you the brown color. Upstream of OCA2, we have a gene called HERC-C2, and HERC-C2 regulates OCA2. So if you're broken in your HERC-C2 gene, you don't make pigment. If you're broken in your OCA2 gene, you don't make this brown pigment. So we have mutations in HERC-C2 and in OCA2 that both give you blue or green eyes. If you make the pigment, you have some form of brown eyes. So where does this live on our planet? This is looking at HERC2, and this is the mutation in it that affects OCA2. And we can see that this HERC2 alleles that cause blue eyes, which it's the color coding on this is very unfortunate, but is orange, compared to the ancestral version, which causes brown eyes, which are blue. I don't know who did the color coding for this. I should probably just remake this graph for them. And we can see that in these populations, we see a large proportion of the brown eyes, and then in sort of some more northern European populations, we see higher prevalence of these blue eyes. Now, OCA2 also rules skin color, but it rules skin color through a slightly different pathway that is regulated by a different gene called APBA2. And so this is looking at the APBA2 alleles. So we have the derived light skin alleles in orange, and then the ancestral dark skin alleles in blue, and you can see where those are on the planet as well. And so we see a lot more of these lighter skin colors in regions way more than we see these lighter eye colors. So lighter eye colors are much more restricted, but lighter skin colors are much less restricted. And we think that that's because there's a much stronger selection on skin color than on eye color. And I mean, I might as well tell you why that is right now, but we're going to look at it again later. But if you have lighter skin, you can deal with less sunlight. So in northern latitudes where there's less sunlight available, your body normally can't produce as much vitamin D. And if you have a melanin layer of protection on your skin, you produce even less vitamin D. So the lighter skin tones in these northern regions allow more UV irradiation to get in onto your deeper layers of your skin, so your body gets told to make vitamin D. But the darker skin colors are protective against getting UV irradiation, against your DNA being broken in your cells from the sunlight. And so if you have enough sunlight around, you're going to have darker skin colors. And so that means there's this really strong selective pressure on skin color that we really don't see for eye color, right? Having lighter or darker eyes doesn't really seem to be beneficial in any particular kind of environment. So other traits are likely to be genetic, but they just haven't even been studied yet. And so things like this include things like eyesight, right? So we know like bad eyesight runs in families, but aside from like degenerative disorders, we don't really know what causes like sort of general nearsightedness genetically. And other things like dimples. I love this dimple picture. Oh my gosh, I love kids with dimples. I have one who has like an almost dimple, and I just found out you can actually get surgery to get like dimples in your cheeks. And I'm like, should I? But it's probably mean he's only 8. I probably should like let him decide if he wants it when he's an adult. They're just so darn cute. So dimples, we know that they probably have some genetic components to them, because we know dimples run in families, right? So I have cousins where their dad has dimples and 2 of the 3 kids have dimples, right? No one else in my family has dimples. Except for my like one son who randomly has like the half dimple. So we know it's like likely genetic, but we don't know what the genes are. Other traits are so highly plastic that their genetic components matter a lot less than the nurture components of this. So some examples, I'll start from this end. Some examples are things like tongue folding. So we know that there's some genetic components to this, right? Like, so there are some people who just don't have like the nervous system and musculature to fold their tongue, but most people can be taught to fold their tongue. So you can learn how, I can't do all these fancy shapes, but most people can at least roll their tongue in half if they learn how to do it. So we know there's a genetic component to it, but the nurture part matters more. If nobody ever told you roll your tongue, you're unlikely to ever learn how to roll your tongue. Things like athletic ability, again, there are absolutely genetic components that feed into athletic ability, including some like really rare genetic components like making a lot of testosterone. That is like super, super useful for having high levels of athleticism, or making more fast twitch muscles than normal. Again, really useful for athleticism. But on the other hand, of course, doing exercise is going to matter most for athleticism. So practice and exercise, those are going to be the main things that are going to drive the variance in athleticism. Over at the very highest end of elite athletes, you need to have both things. You need to have the genetics that, kind of give you higher ability of athleticism, as well as all the practice and training. But you can be on sort of like this high end of genetics, but still be a total couch potato and never have any athletic ability whatsoever, because the nurture matters so much. And it's very similar for intelligence. We definitely know that there is a genetic component to intelligence. If you have really smart parents, you're more likely to be smart. But what matters more than that is what you do with that intelligence. Do you actually study? Do you use different methods to study? Do you learn what the best study methods are for you? Do you practice? When you reach failure, do you look back and reflect and see what made me fail on this task and how can I fix it moving forward? So all of those kinds of things will actually build your intelligence. So if you say got an IQ test when you were 7, that is not at all predictive of how well you're going to do in college, for example, because there's so much of a nurture component here. Like, how much effort are you actually putting into learning the material that ends up outweighing the sort of genetic predispositions to high intelligence. So again, you can really build your intelligence just like you can build your athletic ability. And of course, on the high end, if we're thinking of Newton or Einstein, they're going to have both, right? Not everybody, no matter how much you practice, not everyone can just be an Einstein or a Newton, but we can all get over to the higher end of intelligence than, we might kind kind of initially go into college or go into school, we're thinking ourselves is happening, we can all like move ourselves, can move the needle over to a lot higher than our genetic predisposition. On the other hand, we can also move it to a lot lower. We all know like low effort slackers, right? So people who are really, really smart, they pick up things really quickly, but they don't ever use it. And so their intelligence actually is going to decrease over time. All right, so I want to kind of close out this little mini lecture here, and then I'll do one more on a couple of other genetic traits on how do we connect these genes to traits. So we have all of these traits, like are your earlobes attached or not, and how do we connect them? And we have these techniques called linkage mapping. And in old-fashioned linkage mapping, which I'm not showing on here, researchers would just look at family pedigrees, like we did in previous slides, to see if there are any known genetic traits, so things that we know are mapped to a gene already, that tend to show up in people with the trait under study. So the idea here is that if two alleles of different genes are on the same chromosome, so for example, A and B here, they tend to be inherited together, right? So you tend to get individuals getting big A and big A, or little a and little b, right? So little a and little b tend to travel with each other because they're next door to each other on a chromosome. And the closer they are together on a chromosome, the more often these alleles get inherited together, right? So it's more likely to get a little a with a big C than it is to get a little a with a big B, because they're just closer together on the chromosome. So for example, in fruit fly genes, the genes for wing size and eye color are on the same chromosome. So if you have a fly with small wings and dark eyes crossed to flies with large wings and red eyes, their progeny a couple generations down are less likely to have a combination of the new traits like small wings and red eyes. And that's because in order to mix up those traits, you have to have recombination occurring between those two closely linked alleles. Now, in modern linkage studies, researchers use a technique called GWAS, or genome-wide association studies. And these look at many genetic loci in individuals with or without a trait. So they take individuals with a trait and compare like just thousands of genetic loci to individuals without the trait. And so this is their little comparison tool. It's called a microarray. And these little dots light up different colors, whether you have a particular allele or not. And so they already know the order of all of these genes on the chromosome. And so they can see basically these little dots get made if there are alleles that tend to be found in these ones and that don't tend to be found in these ones. So the higher these dots go up, the more differences there are between these two individuals at that one particular genetic location. And so every time we go above this like little a line that they put here, every time we go up above that, that tells you that's a genetic location that's likely associated with the trait. So that's a genome-wide association study.